Hi everyone, and welcome back to my channel where we talk everything science. My name is Aubrey Molln. I'll be your presenter today, and today's topic is centered around the Renin-Angiotensin-Aldosterone system. Since that's kind of a mouthful, in this presentation we'll just refer to this system as the RAAS. And throughout this presentation we'll also learn not only how it works, but its effects on blood pressure. So without further ado, let's get to it. So jumping right in, it's important to remember that the main job of the cardiovascular system is to keep the blood moving. And to help facilitate that, certain aspects such as blood pressure are very tightly controlled. A major way the body does this is through a set of hormones that help make up the Renin-Angiotensin-Aldosterone system, or RAAS. But first things first, everything starts in the kidneys. So we're going to focus on that. Now, within each kidney, blood from the renal artery flows into the smaller arteries, eventually reaching the tiniest of the arterioles called the efferent arterioles. After the efferent arteriole, blood moves into a tiny capillary bed called the glomerulus. The glomerulus is part of the functional unit of the kidney called the nephron. There's about 1 million nephrons in each kidney, and each of them consists of a renal corpuscle, made up of a glomerulus and the Bowman's capsule, which surrounds it. As, let's see, boom, boom, boom. So here we go. Renal artery, smaller arteries, efferent arteriole, which is this little guy here. This whole unit is called the nephron. Then you start moving into it, and this right here, ta-da, is what you call your renal corpuscle. And then the little kind of red glob in the middle is called a glomerulus, so that's kind of easy to remember. And then here's the Bowman's capsule that kind of creates that C shape that surrounds that glomerulus. Okay, so the renal corpuscle is where blood filtration starts. Interestingly, once the blood leaves the glomerulus, it does not enter into the venules. Actually, the glomerulus funnels the blood into efferent or efferent arterioles, which divide into capillaries a second time. So as you can see here, this little white arrow kind of shows that movement. These capillaries are called the peritubular capillaries because they are arranged around the renal tubule. Now, the renal tubule is made up of the proximal convoluted tubule. The nephron loop, also known as the loop of Henle, as we go back down here, as you see, ta-da, which has an ascending and a descending limb. And finally, you have the distal convoluted tubule as well. So, as you see here, that loop of Henle, your descending limb, your ascending limb, and then, as you can see here, everything's just kind of nicely labeled. There's your renal tubule, there's your peritubular capillaries, etc. Now, as filtrate is formed, it makes its way through the renal tubule, which you can see here. At this point, waste and molecules such as ions and water can be exchanged between the tubules as it makes its way through the descending limb and then the ascending limb, the distal convoluted tubule, and then finally, urine is formed. At the same time, the peritubular capillaries reunite to form larger and larger venous vessels, as you can see here, and then as they reunite, they actually form the renal vein. They take the same exact type of path as the arteries, but in this time in reverse. So as they keep reuniting, they form that large renal vein, which you can see here, which then exits the kidney and then eventually drains into the inferior vena cava of the heart. Okay, so now we're going to actually zoom into the wall of the afferent arterioles, where we're going to find a very special kind of smooth muscle cell called the juxtaglomerular cells, which you can see are these little purple cells right here. Juxta just means next to, and glomerular just means glomerulus. So that just means these particular cells are next to the glomerulus. And the main job of these cells is to always keep an eye open for signals that the blood pressure and or blood volume needs to rise. And these signals can come in 3 different ways through specialized receptors, as you see 1, 2, and 3. So first, the juxtaglomerular cells are actually called mechanoreceptors, also known as baroreceptors. And they're actually designed to mechanically feel if there's low blood pressure in the incoming blood. Now, if they're stretched by an increased blood pressure, they'll actually inhibit renin release. When they are collapsed from low blood pressure, they'll actually stimulate the renin release. Now, second, we'll come over here. Juxtaglomerular cells are supplied by the sympathetic nerve fibers. The sympathetic nervous system is actually activated by these mechanoreceptors stationed strategically in the aortic arch, as you see here, and the carotid sinus to not only measure the immediate blood pressure coming out of the heart, but they actually have other functions as well. They are stretched, then the sympathetic nervous system will be downregulated. However, if they collapse secondary to low blood pressure, then the sympathetic nervous system is activated. Specifically, the sympathetic nerves stimulate the B1 adrenergic receptors on the juxtaglomerular cells to stimulate the renin. Now, if we come over here to number 3, the third signal of the juxtaglomerular cells comes from specialized cells in the wall of the distal convoluted tubule called the macula densa cells. Macula densa cells are actually just chemoreceptors that can sense when glomerular filtration rate increases or decreases based on the quantity of sodium and chloride ions flowing through the tubule. Here's how it works. When blood pressure rises, renal blood flow and, as a consequence, glomerular filtration rate also increases. This means that there's more fluid and more dissolved sodium and chloride ions that reach the macula densa. Now, if the opposite happens and there's decreased fluid and sodium chloride ions getting into the macula densa cells, then that sends a signal to the juxtaglomerular cells to the afferent arteriole. The major signal communicating between the macula densa and the juxtaglomerular cells is the prostaglandins, especially the PGE2. As a result, the use of NSAIDs, or non-steroidal anti-inflammatory drugs, can block this signal and impair the response of the kidney to reduce blood pressure. And all 3 types of signals that I just mentioned here stimulate the juxtaglomerular cells to secrete renin and initiate the RAAS pathway. Now at this stage, renin has been produced. Renin is actually an enzyme that gets into the plasma and it starts to look for its primary substrate, which is angiotensinogen. Angiotensinogen is a very large protein made up of over 400 amino acids that's produced by the liver and is always just kind of hanging out in the blood. When these two meet up, renin actually cleaves off a huge chunk of the angiotensinogen protein, leaving behind only a tiny fragment called angiotensin-1 that's only made up of about 10 amino acids. That's kind of like being a tiny little pearl in a giant oyster. Angiotensin-1 then floats through the blood until it reaches the various capillaries in the body. Now, endothelial cells, which are usually found in the lining vessels of the lungs, have an enzyme on their surface called angiotensin converting enzyme, or ACE for short. This angiotensin converting enzyme actually grabs the angiotensin-1 and chops off 2 more of its amino acids, leaving behind an 8 amino acid chain, which is really a powerful hormone called angiotensin-2. So now we're going to focus on angiotensin-2. Angio just means blood vessels. Tensin just means tense. So that just means that angiotensin-2 goes to smooth muscles lining the arterioles all over the body that contain the angiotensin-2 receptors and cause them to tense up and contract. So if we look here, here's our arteriole. It would actually make these cells on the lining or the inner side lining of the arteriole to contract. This would actually lead to vasoconstriction of these blood vessels and increase the resistance of blood flow. So as angiotensin-2 travels through the blood and reaches the kidneys, it binds to the angiotensin-2 receptors predominantly located along the efferent arterioles. Therefore, it can be said that the efferent arterioles are much more responsive to angiotensin II than the afferent arterioles. So this being said, when there are low levels of angiotensin II, only the efferent arterioles will constrict, and this makes less blood leave the glomerulus. Now, another way to word this is it just makes more blood remain in the glomerulus, thereby increasing the intraglomerular pressure and preserving that glomerular filtration rate. Now, on the opposite end of the spectrum, when there is higher levels of angiotensin II, both the afferent and the efferent arterioles constrict, and this decreases both renal blood flow and glomerular filtration rate. Angiotensin II also acts on the cells that line the proximal convoluted tubule, making them reabsorb more sodium ions from the filtrate. And by the process of osmosis, water begins to follow the sodium ions. Furthermore, angiotensin II also acts on the hypothalamus, which is located at the base of the brain, as we see over here. This is where it stimulates thirst and increases the production of the antidiuretic hormone, or also known as ADH. ADH has its main effect on the cells lining the distal convoluted tubule and collecting duct, where it actually helps to stimulate more aquaporins to get into the luminal surface of these cells. As you can see here, here's that distal convoluted tubule, and then here are those aquaporins that help with the movement of water. These aquaporins make these cells more permeable to water, which increases water absorption. Ultimately, the increased thirst and water reabsorption increases the total circulating blood volume or blood flow, which if we look down here can be symbolized as Q. In addition, the increased peripheral arterial resistance increases the total body resistance, which we can represent by R. Since the change in blood pressure P over here equals the blood flow Q times R, the blood pressure will rise. But angiotensin II's work isn't over yet. It also helps stimulate the adrenal glands, which are a pair of glands located on the upper side of the kidneys. Here's your kidney, here's your adrenal gland. If we take a closer look at the adrenal gland and then zoom in on a cross section, we'll see that it's made up of two main parts. These parts include the inner medulla and the outer cortex. The cortex is further subdivided, so we're looking at that outside kind of yellowish rim here. It's actually going to be subdivided into three more layers: the zona glomerulosa, the zona fasciculata, and the zona reticularis. Now, the angiotensin II stimulates cells in the outermost layer, which we're going to focus on up here. And that is the zona glomerulosa, which secretes a hormone called aldosterone. So what really is aldosterone? Aldosterone belongs to a class of steroids or lipid-soluble hormones called the mineralocorticoids, which helps regulate the sodium balance in the body. Aldosterone does that by acting on the cells that line the distal tubules and collecting ducts, as you can see kind of pointed over here. Now, aldosterone gets into those cells and then binds to the receptors, forming an aldosterone receptor complex. This complex is able to control the expression of genes, which can increase levels of certain proteins over others. One protein in particular that gets expressed a lot more often due to this connection is the sodium-potassium ion pump, which sits on the basal surface of the cells. These pumps actually push sodium into the blood while pulling potassium ions into the cell. Ultimately, this helps to drive a concentration gradient that pulls sodium and water out of the lumen of the tubule and into the blood and secrete potassium. In fact, most patients with intensive activation of the RAAS system can be profoundly hypokalemic. As before, this helps to boost the blood pressure. Now keep in mind though, that this process can take several hours or even days. So honestly, it's considered a late response of the RAAS. All right, so that was a lot of information that we just went through. As a quick recap, Whenever there's a decrease in blood pressure as detected by the baroreceptors of the carotid sinus or the aortic arch, or the juxtaglomerular cells, the sympathetic nerves getting stimulated, or even the macular densa cells sensing less sodium and chloride ions flowing through the tubules, the kidneys, as we come back up here, will actually secrete renin that converts to angiotensinogen, and then to angiotensin 1, and then angiotensin 2. After that, the angiotensin converting enzyme converts the angiotensin 1 to angiotensin 2. Angiotensin 2 causes the efferent arterioles over here to constrict more than the afferent arterioles, which then increases the glomerular filtration rate. It also causes the proximal tubule to reabsorb more sodium ions from the filtrate. It increases thirst, and it helps increase blood pressure, while also stimulating the adrenal cortex over here to release aldosterone, which gets into the kidneys to help them retain sodium and water. So now that we've kind of concluded the meat of the presentation, I want to take a few moments to talk about a scholarly article that kind of applies what we've just learned. This article is called the Renin-Angiotensin-Aldosterone System, a specific target for hypertension management, written by Wer and Zhao. And they actually tested ways to help either block or stimulate the RAAS system to help people manage their high blood pressure because it has direct impact on blood pressure. Not only that, but they also took their findings and compared it to ACE inhibitors, which we've talked about in this presentation, in patients. So basically they had patients that had ACE inhibitors and patients who had the angiotensin 2 blockers. And they actually found that it was quite beneficial on both aspects. But a big thing that they really noticed in their clinical trials, that angiotensin is an excellent target for pharmaceutical blockage, and it also helped reduce patients' withdrawals from drug therapy. The whole article was very interesting, so I definitely recommend a good read through it. It shows their different approaches on how they manipulated the RAAS system and basically how they applied it. Now, this article was written in 1999, so a lot more advances has happened since then. But this was a very good preliminary research project with clinical trials in play that I definitely recommend if you are curious to dive a little deeper into the topic. And that is all I have for you today, guys. Thank you so much for coming out and listening to this presentation. Again, I hope that you learned something or can take some information away from this presentation. For those of you who are curious and want to go a little bit further and read about those clinical trials I was just talking about, this would be the article for you at the very bottom here. Again, a great article, great research and clinical trial type application of this information. So thank you again for being here today, and I hope to see you again. Until then, have a beautiful day.